So obviously the main use for carbohydrates is that they are the source of energy. Okay. We call them carbohydrates because they are formed by carbon, hydrogen, and oxygen. And another term for carbohydrates that we sometimes use is saccharides. So the main source of carbohydrates, well, the original source of carbohydrates, is the process of photosynthesis. Plants use carbon dioxide, water, and energy from the sun to synthesize glucose. Now, so this is shown right here. In photosynthesis, we take two inorganic compounds, carbon dioxide and water, and using energy of the sun and some electrons, make glucose. Now, in the process of respiration, not only respiration, there are other processes that animals can use, but generally speaking, in the process of respiration, glucose is oxidized by oxygen back to carbon dioxide and water, releasing energy. So this is one of the main components of the cycling between organic carbon in carbohydrates and inorganic carbon in carbon dioxide. The terms that show us how many building blocks are in the specific carbohydrates are monosaccharides, disaccharides, sometimes trisaccharides, and polysaccharides. Now, from myself, I will add something as well, okay? So, another term that we can use is oligosaccharides, okay? Oligo means a few. So, for instance, if you have one molecule of monosaccharide, we call it one molecule of carbohydrate, say glucose, we call it a monosaccharide. We combine two molecules of glucose together, that would be a disaccharide that we will call maltose. We combine 5 together, and strictly speaking, we would call it an oligosaccharide. Now, if we combine, 50 molecules of glucose, that would be poly. Mono, tri, oligo, polysaccharides can all be hydrolyzed. You can see it here. Addition of water breaks them down into individual building blocks. Okay? Any questions so far? Okay. Monosaccharides. 3 to 7 carbons. 1 carbon will always be a part of a carbonyl group, whether it is an aldehyde or a ketone. All other carbons have hydroxyl groups on them, as you can see in these examples. And the classification of monosaccharides is partially based upon whether they contain an aldehyde or a keto group. If a monosaccharide contains an aldehyde, we will call it an aldose. And if it contains a keto group, we'll call it a ketose. So at the end of the day, to describe any specific monosaccharide, we will characterize it as aldose or ketose, and we will also describe how many carbons comprise the molecule. So the classification based on the numbers is triose, 3 carbons. You just sew a triose, erythrose. Tetrose, 4 carbons. Pentose, and hexose. In our description, we can combine these 2 categories. So if we have a monosaccharide with 5 carbons that contains an aldehyde group, we will call it an aldopentose. If we have a monosaccharide with 6 carbons that contains a keto group, we're going to call it a ketohexose. So these are some examples of different types of monosaccharides. For instance, glyceraldehyde has 3 carbons and contains an aldehyde group. We're going to call it aldotriose. Thereose contains 4 carbons and also aldehyde group. We're going to call it aldotetrose since there are 4 carbons. Ribose, a monosaccharide that is used in building of RNA molecule, is an aldose that has 5 carbons, therefore aldopentose. Fructose, a monosaccharide that contains 6 carbons and a keto group, will be ketohexose. I think it's pretty straightforward. So let's see if we can identify these monosaccharides as basically give it a characteristic. Okay? Let me see if I can write it down. Okay. I wonder how I am. Let's see. How do I insert a text? Well, what is A? We'll have to say this probably. How many carbons, first of all, in the A?
6.
So 6. Aldehyde or ketone? So aldohexose. Yes. I'm desperately trying to figure out how to type something. Well, I will have to write it down this way. So aldo hexose. Right? Now, what about second one? What type of carbonyl is that? Ketone. How many carbons?
5.
So, ketopentose. Correct. Yes, very good. Now, normal blood level of glucose, what's considered to be normal blood level, is 70 to 90 milligrams per deciliter. Technically speaking, anything up to 100 is considered to be normal. Okay? People who have diabetes is classified as the fasting blood glucose above 125 milligrams per deciliter. Okay? Now, to test it, to test whether or not a person has diabetes or pre-diabetic state, the glucose tolerance test is performed. So, as a person who actually did perform glucose tolerance test, not on humans but on mice, so some of that stuff is not entirely correct in terms of the graph. Here's what happens. You are given a glucose solution, a certain amount per kilogram. I don't remember what are the numbers. And then your blood glucose levels are tested usually 15, 30, 45, 60 minutes, 90 minutes, and then 2 hours after the ingestion. You have normal glucose tolerance if after 2 hours your glucose levels go back to normal. Okay? Hypoglycemia are low glucose levels, like very low. It's not entirely correct here, showing that insulin just knocks you out pretty much. And then hyperglycemia here, what you see is basically diabetes. Okay, fasting glucose is high, and it doesn't go back to normal levels in 2 hours. So there are two types of diabetes, as you know. Call it diabetes mellitus. Type 1 diabetes is when the pancreas doesn't produce enough insulin. This is an autoimmune disease that is characteristic for starts usually is diagnosed when we are kids, okay? And it is of an autoimmune origin. Now, another type of diabetes, as you may be aware, is type 2. In type 2 diabetes, usually people are diagnosed later in life, in the 20s, 30s, whatnot. And for the most part, it is a result of dietary and other lifestyle choices. So, consumption of high carbohydrate food with a lot of simple sugars results in the constant release of insulin and makes cells in your body, mostly liver cells, resistant to insulin. So, although your pancreas produces enough insulin based on the standards, liver becomes non-responsive, and people have elevated plasma glucose levels. Now, 350 milligrams per deciliter, pretty average levels for a person with uncontrolled diabetes. It can be up to 800 milligrams per deciliter in a person who does not, like, say, type 1 or type 2, who eats high carbohydrate meal. Okay? Symptoms: thirst and excessive urination. Now I'm going to give you some physiology link. People become thirsty and urinate a lot when they have diabetes because their kidneys try to get rid of all the excess glucose. And in order to get rid of it, they have to flush it, almost literally flush it out of the system, and people will lose water. When they lose water, they're thirsty, okay? Now, you may find it counterintuitive that people have increased appetite and weight loss. Well, not in type 2 diabetes usually, because it's associated mostly with obesity as well, okay? But if you think about it, your cells cannot take glucose out of the bloodstream. Am I making sense? So if they cannot use glucose, they have to use something else. What is the second source of energy in humans after carbohydrates?
Protein.
Not a protein. Protein is the last third. First is carbohydrates. Fat. People will start losing fat. I mean, I'm not suggesting that giving yourself type 1 diabetes is a reliable way to, lose weight, but okay. So hypoglycemia, when people have very low blood sugar, okay. Pancreatic tumor sometimes may result in overproduction of insulin by pancreas, okay. Usually it is that. And when you don't have enough sugar, you have dizziness and muscle tremors. Reasons for it: when your muscles don't have enough don't have enough glucose, they can't produce enough ATP, and that sends muscles to a contracture. Okay? So basically, contractions. Now, why dizziness? Your neurons completely rely on glucose as a nutrient. If you don't give your neurons enough glucose, they're going to stop working, and you can actually pass out. So far, we're good? Okay. So, what are the chemical properties? Well, we're going to talk about oxidation and reduction for the most part. So, monosaccharides in the solution have so-called open chain form, like this, okay? So this aldehyde group, if it's an aldose, can be oxidized to an acid, and this picture of monosaccharides is used to determine them in the solution using Benedict's test. We talked about Benedict's test when we covered aldehydes. Okay? In Benedict's test, you have blue solution of the copper 2. When copper 2 is boiled with an aldose, such as glucose, it oxidizes glucose to a gluconic acid and produces partially reduced copper 1 in the form of copper 1 oxide, which has a brick red color, and it's a very cool test. So the acids that are produced are called sugar acids, and their names are formed by reducing OSE, for example in glucose, with onic acid. So if it's a glucose, we're talking about gluconic acid. If it is erythrose, we can say erythronic acid. Now, fructose is ketohexose. It contains a keto group. If you look at a keto group here, usually it's not oxidized. It can't be oxidized. It can't be oxidized because it is connected to two carbons rather than to a carbon and a hydrogen. However, in fructose, keto group is located at the carbon 2. So fructose, when it is tested with the Benedict's reagent, can rearrange its carbonyl group, be converted into glucose, and glucose at this point can be oxidized to gluconic acid. So in a basic, and that's important, in only that can happen only in the presence of base, only with relatively high pH. Okay? So this is what happens. On the left, you can see a D-fructose, a ketose. On the right, you see the glucose, which is aldose. Now, aldose at this point can be oxidized by copper to gluconic acid. Now, what about a reduction of monosaccharides? So you can use hydrogen and platinum catalyst to reduce D-glucose, an aldose, a sugar, into a sugar alcohol called D-glucitol. Okay? So another term for D-glucitol is D-sorbitol. And the use for these sugar alcohols, which can be derived only from aldoses, only from so-called reducing sugars, okay, they're used as sweeteners. So glucose, obviously, is sweet. When you reduce glucose to glucitol or sorbitol, it's also sweet. But the good thing is, it cannot be fermented by microorganisms in your mouth, okay? And it is safe to consume for people with diabetes. Okay? Am I clear? So, naming the OSE is replaced with ITO. For instance, xylose, when reduced, produces xylitol. Mannose, when reduced, produces mannitol. Okay? Now, why it is important that those sugar alcohols are not fermented by the microorganisms in your mouth? Because if they would be, you would have acid formed in your mouth, and that would negatively affect your enamel. In this case, in a sugarless gum, it is safe. It is safe to chew it. Okay? So, people with diabetes, as I mentioned before, people with, importantly, uncontrolled diabetes, people whose blood glucose levels are very high, have a lot of glucose in the urine. Am I making sense? Now, that happens when the blood glucose levels exceed 160 milligrams per deciliter, which is very easy for a person with diabetes, okay? And when that happens and glucose ends up in the urine, we call this condition glucoseuria. So, glucose in the urine can be tested by a paper strip. Don't remember, did we already work with fake urine? We didn't? No. We're going to work with fake urine. So basically doing the urine test, urinalysis, okay? So what happens in this testing, you have glucose, and there is an enzyme in the strip called glucose oxidase. In the presence of oxygen, it oxidizes aldehyde to carboxylic acid, producing gluconic acid. And the byproduct of that reaction is hydrogen peroxide. Hydrogen peroxide oxidizes the dye that's added to the strip to methylaniline, and that oxidation results in the production of colored product, and you can see the band. And the intensity of the band will correspond to, how much glucose there is. Okay? Now, to be honest, I think much easier test for glucose is just to do the little prick, you know? That makes sense? Like if you suspect that your patient has diabetes, just do the standard glucose test using glucometer, okay? This is used more for, like when you give urine for urinalysis, they will also determine whether or not you have glucose. Now, I'm not going to read all those percentages for you, okay? You can read the table yourself. I have another question. Obviously, those test strips and glucometers were not known to physicians, say, early in 20th century. Would you agree with me? Yeah? So here's my question. Early 20th century, you're a doctor. You need to test if your patient has glucose in the urine, if your patient has diabetes. What would you do?
The taste. They use the taste.
Taste, yes. Taste the urine. There is another test. Will work in the summer, not so much in the winter or fall.
Sweat test.
What do you mean?
I have no idea.
No, like you have a sample of urine, okay? It presumably contains glucose, a lot of glucose. You don't want to taste it.
Does that have any with ants or insects? No.
No. You have glucose, you have urine, you have sweet urine.
Mm-hmm.
Sweet things, what do they attract?
Birds.
Flies or
Yeah, that's what I
wasps?
said.
Huh?
That's what I said.
Oh.
Ants and
Okay.
all that.
Yeah. Doctors would put a little, like plate with urine on the windowsill, and we'll see. If there are flies, there's a pretty good reason for it, the glucose. Now, when you combine two saccharides, obviously you form disaccharide, okay? The examples of disaccharides that we're going to talk about are lactose, sucrose, and fructose, okay? Lactose, sucrose, and maltose, my bad. So this table is what you need to know about disaccharides. You do need to know them. So first disaccharide is the combination of two glucose molecules. We call it maltose. It's found, in the malt. The combination of glucose and galactose give you lactose. And the combination of glucose and fructose give you sucrose. I want to highlight something for you folks. These reactions, the product in these reactions is disaccharide and water. We call those reactions of dehydration. So maltose, composed of two alpha D glucose molecules. We're going to figure out. So I will have to quickly, crash course, explain the idea of chirality and ability to rotate light. Okay. So look at your hands, please. Seriously, right now, look at your hands like this. Can you completely overlay them like this? You can't, right? Your hands are mirror images of each other. Am I making sense? They not, basically 5 fingers and everything, but they're not identical. When you have two molecules that are mirror images of each other, yeah, you can't, you have to, there's no way, even if you chop it off, there's no way, okay? They're not identical. Your thumbs are looking in different directions. So when you have two molecules that are exactly mirror images of each other, those molecules are chiral, kairos in Greek, is hand. And depending on how these molecules are organized, I'm being extremely simplistic. They are given a letter prefix, D or L. Am I making sense? It depends on in which direction they rotate polarized light, whatever, physics, doesn't matter. But why this is important? Listen, all carbohydrates in the nature, lactose, starch, cellulose, whatever, all of them consist of D monosaccharides. Does that make sense? Like D is a very specific characteristic, like in which order the groups are arranged, okay? Does that make sense to you? Okay? Now, alpha, when we say alpha carbon means that the OH group faces down on the first carbon, okay? If it faces up, it's beta. Does it make sense? Good? So here you have two OH groups. Both are alpha OH groups. One OH group on the left is attached to the first carbon. Okay? Another OH group on the right, so first carbon, another OH group is attached to the fourth carbon. Therefore, we say that two molecules of glucose and a molecule of maltose are connected by alpha 1 for glycosidic bond. Does it make sense? This is really important because you will see the big difference when we get to cellulose. So far, is that clear? Can you digest maltose? Dude. Cereals, candies, beer. Can you digest? Yes, you can digest maltose. Okay? Lactose is the product in which galactose and glucose are combined. Okay? So you have alpha glucose and you have beta D-galactose. Okay? So since galactose is beta and the bond goes from 1 to 4 in that direction, we say what is the position of OH group at the first carbon. It's beta, you see it faces up. Am I making sense? So we call this bond beta 1,4 glycosidic bond. Can we digest lactose? Yes. And yeah, lactose in human milk and lactose in cow milk, same lactose, same sugar. A piece of trivia, milk of which animal, composition-wise, is closest to human milk? It's a trivia. You either know it or you don't. It's not a logical answer. What do you think?
I was going to say cow milk.
Cow? No. Cow? Huh?
Goat.
Nope. Whale.
What?
Don't ask me how they milked it, but it was shown whale milk is the closest to human milk. So the bond, as we mentioned before, is beta 1,4 glycosidic because the OH group on the first carbon of beta D-galactose is in the beta position. That's your beta position. Am I making sense? Okay, awesome. Sucrose. So where do we get it from? Well, I believe in the United States, sucrose is produced from sugar beet. Okay, sugar cane, as far as I know, is sort of more efficient. This is why, like Brazil and Cuba are one of the biggest producers of sugar in the world. Okay, and it consists of alpha D glucose and beta D fructose. Now the glycosidic bond is different. Look at this. So you have the bond, remember fructose is the ketose. You have a bond between hydroxyl group on the first carbon, which is in alpha position, and hydroxyl group on the second carbon of fructose, which is in beta position. Okay? So we call it alpha beta 1 2 glycosidic bond. Okay? Now, sucrose is not a reducing sugar since it cannot form an open chain. So it does not produce Benedict's reagent. Now, a little comment on a sucrose. Hear me out. When you eat something sweet, something sweetened with sugar, sugar is converted in your mouth by your oral bacteria. It's broken down into fructose and glucose. And your oral bacteria ferment fructose, producing acid, and that causes dental caries. Does that make sense? Specifically sucrose, not lactose, not even maltose, but sucrose. They ferment fructose. So in order to replace sugars, we use artificial sweeteners. The comparison is set to sucrose and the value of sweetness of sucrose is 100. You know, the reference point. So this table kind of summarizes what you're going to see. So you see lactose and maltose aren't as sweet, which you can, like milk itself isn't sweet. Beer, which contains a lot of maltose, isn't sweet. Okay? And your sugar alcohols aren't particularly sweet as well. When you start looking at the sweeteners that are not carbohydrates, it goes pretty insane. Okay? So, I have a question for you folks. Yeah, go ahead. Question? So, these are typical sweeteners that you're going to find in your local Starbucks, you know, like aspartame and stevia and sucralose. Question: are they bad for you? Sweeteners.
Yes.
The dose makes the poison. So my point is, if you replace sugar with sweeteners, overall, you're going to benefit. The amounts of sweeteners in drinks like, I don't know, Diet Coke are so low because they are so sweet, the sweeteners, I mean, that the dose that you receive is minuscule. However, it is not really clear how consuming something that's sweet but doesn't contain sugars, how does it affect the behavior of your gut and microorganisms in your gut? Because you're pretty much cheating. Does that make sense? Your taste receptors tell your brain, hey, it's sweet. It gets in your intestines, and there's definitely communication between the brain and your gut. It gets in your intestines, and suddenly there's nothing to digest, no carbohydrates. Make sense? It's really the big question. So this is sucralose. If you look at this, it kind of looks like sugar, but there's a chloride here. So, and there's a chloride here, and there's a chloride here. So by replacing this, it's not a sugar anymore. Even if it's broken down, it's not broken down into glucose and fructose. Okay? So it doesn't provide any energy for your body because your cells cannot metabolize it. Now, sucralose is pretty much a sweetener that is based on carbohydrates. Am I making sense? Like, structurally, it's pretty similar. Now, aspartame is not a carbohydrate-based sweetener. Can you see that? It's pretty obvious, right? Does that make sense? It's pretty much a combination of two amino acids, okay? Aspartate and modified phenylalanine. So, lactose. Which monosaccharides form lactose?
Lactose.
Galactose and
Glucose.
glucose. Remember, there's always glucose. There's always glucose. And for lactose, think galactose. Maltose.
Glucose and fructose.
Nope, that's sugar.
Oh.
That will be,
Oh.
okay, remember, maltose is purely glucose.
Oh, two glucose.
Two glucoses, yes. Yes. Okay, polysaccharides. Well, obviously, many individual elements. And we're going to learn the structural differences between starch, glycogen, cellulose, and I think we're talking about chitin. So, starch, glycogen, and cellulose are all formed out of glucose molecules. You may ask, hold on a second, what is the difference? The difference is how these glucose molecules are arranged and connected to each other. Okay? So, first, starch. Okay? So you can see that in the molecule of starch, individual glucose residues are connected by alpha-1,4 glycosidic bond. And when these chains of glucose molecules do not have branches, we call it amylose. Okay? In amylopectin, we have two kinds of bonds. One is classic alpha-1,4 glycosidic. Another one is alpha-1,6 glycosidic bond, which connects first carbon of one glucose and sixth carbon of another, and allows this molecule to branch. When you eat any starchy food, you eat both those things. So starch contains amylose and amylopectin. So amylose makes up about 20% of starch. Okay? And it's a straight chain polymer. It's called straight chain because there's no branching. In reality, they are twisted. You can see it on this image. Okay? You can see that amylose is a helix and amylopectin is not. Okay? And it contains only alpha-1,4 glycosidic bonds. Amylopectin is 80% of starch, the branched one. Okay? And as we mentioned, contains alpha-1,4 and alpha-1,6 glycosidic bonds. Alpha-1,6 bonds are responsible for branching. We're good so far. I can leave it there for as long as you need. All right? Now, glycogen. So, starch. Where do you find starch? Tell me.
Like where in the body?
No. You can't find starch in the body. It gets digested.
Like food?
Food, yeah.
Yeah.
Which food?
Like grains, bread?
Bread. So starchy food, right?
Right.
Potatoes, rice, right? So glycogen, so those foods, where do they come from? Animals or plants? Plants. Starch is a uniquely plant product. Okay? And if you think about it, so let me ask you this. Why do plants produce starch? What is the purpose of starch in plants? Let's start with, why do we eat starchy foods?
It's salty.
Say again?
I was gonna say salty, but
To get what do we get out of starch? What is it used for in the body? As energy, yeah. It's a source of energy, right? So what is the purpose of starch in plants? Energy, yeah. Anyone ever grew a potato plant? No? Yes. Let's say, like, you collect the tubers, whatever. It's not actually a root, it's part of a stem. But you collect those potatoes. Now imagine you have the plant, and instead of collecting, you just leave it. So winter comes, the top of the plant dies, okay? What's going to happen next year at the same spot?
That will clear up again.
You're going to get new plants from the tubers that are sitting in the ground. What's going to happen to those potatoes in the ground as the plant grows? They're going to shrivel because the starch will be used as the source of energy initially. Does that make sense? So, same story. Glycogen is the source of energy. It serves the same purpose but in animals. Glycogen is branched polysaccharide, okay? It is stored in humans in the liver and the muscle, okay? And when we need an immediate supply of glucose, glycogen can be hydrolyzed and released, and glucose will be released from cells into the blood. And other tissues, let's say brain that cannot store glycogen, will receive its glucose and we're fine, okay? It's more branched. Other than that, the glycosidic bonds are the same. Alpha 1,4 glycosidic bonds, responsible for the formation of chains, and alpha 1,6 glycosidic bonds are responsible for branching. Good? Make sense? Why I paid so much attention to the fact that in the molecule of amylose, or in the molecule of glucose, in the molecule of maltose, two glucose residues are connected to each other by alpha 1,4 glycosidic bonds. Because when we start looking at the cellulose, turns out that it's a long chain of glucose residues, but they are connected not by alpha 1,4, but by beta 1,4 glycosidic bonds. Okay? Does it make sense right now? So, some statements here are plainly wrong, and I will highlight which statements are. For now, can you show me any material on your desks that contains or made of cellulose? Go on.
the top.
No. Show it to everybody. Yeah? Paper.
Ah,
Paper. Now, can you digest paper?
yes.
No. You can eat it, you can't digest it. I mean, I don't recommend. Tastes, there's no taste. But, you know, when people you can take a sheet of paper, you can eat it.
So what happens to it after that?
Well, guess what? Comes out.
Oh, okay.
It comes out, I wouldn't say untouched. It's going to be
Yeah.
in a pretty bad shape, but it's not going to be broken down. Another,
It's not like corn.
no,
Corn doesn't come out. Well, it comes out. Yeah,
well, Corn, some parts of a corn, you're talking about corn kernels? Yeah, you can digest them. There's a lot of starch in them. The example of a food that is pretty much cellulose, celery. I share your sentiment about celery, but celery has almost no digestible parts in it. But when you eat it, it serves as a brush in your, like, brush, quote unquote, in your intestines. Like if you have constipation, it's good. It's going to move things, That make sense? Right? Now, cellulose, the authors here say that they cannot form hydrogen bonds with water. So, it's wrong. If I will ask Taylor to open her bowl and pour water on a notebook,
I'm not gonna.
would it get wet, the notebook? Yes. So cellulose can form hydrogen bonds with water. Otherwise, plants wouldn't be able to deliver fluids from the ground up the stem. Am I clear? Starch isn't soluble. Try to dissolve a piece of bread. You see, they claim that it's insoluble. Starch and cellulose are not soluble in water. You understand what I'm saying, right? But the role of cellulose is that it forms cell walls in plants, and this is why we have trees, effectively, because of the cellulose in the cell walls. Now, resistance to hydrolysis, that's questionable, but this statement is absolutely true. Humans do not have enzymes that break down these beta-1,4 glycosidic bonds. That makes sense. Now, can you name an animal that can eat and actually digest plants?
Goat.
Sure. Goat would work. Okay. Is there a difference between a goat's stomach and a human stomach? Goats can digest cellulose, human can't. Is there a difference? It's a leading question, folks.
Yes.
Yes. And the big difference, the ruminants, like goats or cows or horses, have several chambers in their stomach, and they regurgitate food. So the food goes into the stomach, gets partially digested, they regurgitate it back in the mouth. Basically, they vomit in their mouth deliberately, chew it again, swallow it back. It takes several hours for the food to spend in the stomach of a goat or a cow to be fully digested. And one of the leading roles in this process is taken by bacteria that live in those intestines. Now, polysaccharides and the types of glycosidic bonds in each. So what do we have on the left? What is that? What polysaccharide? If it's cotton. Can you eat cotton? Can you digest cotton? You can. You shouldn't. You can eat cotton, but you can't digest cotton. So if you can't digest it, is it a cellulose? Cellulose. Okay. Cellulose, perfect. Type of bonds, beta. Okay. What about that on the right? What type? of polysaccharide in potatoes?
Starch.
Yes, it is starch.
Hey. I got it.
And type of bonds?
Amyloses or
Alpha, right? 1,4,
And 1,6.
glycosidic. What was your...
Is it also alpha 1,6?
Excellent. Thank you so much. Alpha, I forgot about those. 6, glycosidic. So the question, alpha 1,4 glycosidic, in which components of starch is it found? Amylose, yes, and
And glycogen.
amylopectin. There's no glycogen in potatoes.
Oh, wait.
It's okay. Alpha 1,6 glycosidic, only amylopectin. Does that make sense?
Yeah.
Yes, good. So